रुकैया परवीन, रुकैया परवीन।
Welcome back to the post-lunch session. Hope all of you had a sumptuous lunch. Please call your friends who are outside to come into the hall. And those of you seated behind may kindly come forward. It will open here also. I have to read. Kindly come forward. Kindly come forward.
All are requested to please come ahead. Hello, students. Silence, please, silence. All those who are sitting at the back bench, please, it is earnestly requested, come forward. Please, students sitting at the left side, please come forward. Please come. I am requesting you. Please come ahead. Don't gossip.
Dr. C.P. Pandey and Dr. Tirumurugan Murli. May I request them to kindly come on to the stage, please? Dr. Malliga is Assistant Professor, College of MLT, MTPG and RIHS Puducherry. She is also an alumnus of JIPMER and has a PhD in Pathology from SBV University. She was a resource person in many conferences and she has presented many scientific papers. She has organized many programs for celebration of NMLPW celebrations and organized few medical camps also. She has been a part of the organizing committee. Welcome, Dr. Malika. Next chairperson is Dr. C.P. Pandey. He is Principal Technical Officer, CSIR Central Drug Research Institute, Lucknow. He is also the Manager of Science and Technology Facility with more than 35 years of experience in a broad spectrum research and development, namely holistic health systems, diagnostics, drug discovery, facility creation, and human resource development. Welcome, sir. And third chairperson is Dr. Tirumurugan Murli. He is presently working as Principal, College of MLT at Mother Teresa Postgraduate and Research Institute, Puducherry. His passion for teaching is for more than 23 years and was duly recognized by conferring him with Dr. Radhakrishnan Award. He was also awarded the Mother Teresa Award and a certificate of appreciation by Sri Aurobindo Society. He has also got a certificate of commitment for his active involvement in the COVID pandemic. He was also recognized as Pillars of Puducherry for playing the role of enabler. He was also the recipient of ICMLS Academic Excellence Award 2023. He has several publications to his credit and has been a resource person in innumerable conferences and has been organizing state-level conference for the medical laboratory professionals and students annually since 2015. Welcome, sir. So the first talk is laboratory automation. Where are we? And to give the talk, I would request Dr. Surendra Kumar Sharma, Pijimar, Chandigarh, to kindly come on to the stage. Sir, requesting Dr. C.P. Pandey to kindly introduce, sir.
Good afternoon, all of you. The toughest session in any conference is the post-lunch session. It's both for the audience as well as for the speaker, but for the audience to be alert and the speaker to keep them alert. But I'm pretty sure that the speakers, the vibrant speakers here are going to make it very enjoyable for you to hear to them. He's a senior medical technologist in the Department of Experimental Medicine and Biotechnology, PGI, Chandigarh. Has more than 25 years of experience, has a lot of publications in reputed indexed journals, both national and international. He was the recipient of the Best Oral Presentation in the International Forum in the conference organized by the Korean Association of Medical Technologists. He was a technical inspector and assessor for inspection of affiliation of nursing and paramedical colleges in Uttar Pradesh, a subject expert for medical lab sciences. And of course, to add to it, we need to add that he's also a recipient of the prestigious ICMLS Young Scientist Award, which he received today in this August gathering. I'm happy to invite Dr. Surendra Kumar Sharma for his talk.
Thank you, sir. Thanks a lot. A very good evening, all of you. I know this is a difficult task for me as all of you just had your lunch. So today I'm going to talk regarding the total laboratory automation and where we are standing globally. As you all know that total laboratory accreditation automation is associated with reduced human errors and consistency with precision that robotics and computers can play. Total laboratory automation solution helps to achieve elevated role in quality in the future of ever increasing levels of automation as well as accreditation. Since morning we are listening about the total laboratory automations, lab information management automations as well as accreditations. So total laboratory automations is going to help once we are having a very large number of samples. or biochemistry, as well as few very special investigations. In those areas, total laboratory automation is not feasible, and we do have the automations with different standalone analyzers. As you know, the automation lines covers the pre-analytics, chemistry, immunoassay analyzers, hematology, as well as post-analytical refrigeration storage unit. So overall, the whole process is human-free. So once we have scanned the samples, the instruments is going to be selfly analyzed for which investigations the samples has been received, and they perform the investigations through the TLA. And it is having an added advantage, like it is storing the samples by himself, so there is no need for the human retrieval. So once we have processed samples and we need to repeat the samples, in that time one has to go deep freezer to take all the samples out, and it is really difficult to segregate the particular samples that we need to be processed. So TLA helps in that solution of post-storage also. So it essentially have 4 sub-processes. First is transport of samples to the laboratories, pre-analytical processes, transport of samples to analytical instruments, and then intermediate storage and samples disposals. So majority of the errors area is pre-analytical. So nowadays we do have the instruments, even for the blood collections, the fewer companies are introducing their instruments, those can collect the samples by themselves. So still we do have expertise in the phlebotomy. So here is a classical example of one of the TLA laboratories. So here you can see all the lines are working. Samples received, they are identified, if required they centrifuge by themselves, they do the allocating, they do hematological lines, chemistry, immunohistochemistry, microbiology, urinalysis, and then finally sealing and sample storage if required, otherwise disposing. So this is a overall TLA. These are classical examples. All the instruments are covered with the TLA lines. This is another company, AutoBio, which also provides the TLA solutions as per the requirement of the laboratories. Here you can see all the instruments are aligned through these samples lines as per our requirements, as per our areas, as per where we are getting the higher number of samples. Those instruments can be connected with these particular lines. They do the quality control sampling. All data is available round the clock. Wherever we get any error, here you can see directly the samples are put it in that automation line, and it segregates itself where it's supposed to go. It labeled himself, then it reaches to particular instruments, they perform uncapping of the samples, sealing of the samples, and through these, they are going through these all automation lines. So it's totally human free. One has to sit on the system and see all the quality charts, how the instruments are working and how the results we are getting on the TLA. Major private laboratories are already having the TLA solutions in those areas where they are getting very high number of samples. Say they do centrifugation if required, otherwise they bypass that instrument directly. Through these TLAs, accreditation is very much easier because each and every sample processing, their TAT is also maintained. It is reducing the time of reporting the samples, And these are successful only in those laboratories where we are getting very high number of samples. Probably AIIMS is having the smart lab. They must be having this kind of instruments here. So, standalone laboratories used to have the automation. This is a second group of laboratories like therapeutic drug monitoring, endocrine and hormone testings, various vitamin testings, and various pharmacological laboratories used to have the automated instruments. So, we need to prepare our own laboratory development methods, and we used to use the automation for those areas. TLA could not help in those areas where the medical technologists are supposed to develop a laboratory-based testing. In these laboratories, only the few pre-analytical areas can only be automated, like the sample processing up to the tray loading. TLA has benefited central processing lab in cost containment, efficacy improvement, and most importantly, quality control, patient care. So this is about that, and last month I had visited one of the TLA laboratory in Korea, in AMLS, madam must have gone. So I would like to share that videos of that, my visit of that total laboratory automation in the ION laboratory of Seoul. Here you can see a hematology laboratory. It is a fully automated hematology laboratory. They are running a TLA, as well as they do have the robots to process, to bring their stat samples, because a technocrat is really difficult for those countries where the manpower costing is very high. So for them, it is a solution for them. Here you can see. This robot is coming to taking stat samples. Can we have the audio in this? Audio?
Through the Bluetooth, so it can move the floor of the building. Most of them, we have two robots.
So these are their robots basically.
It's very busy.
And they were bringing their samples. You can see the number of samples they were receiving.
Over there, you can see the dashboard. We use that, we check the progress of the situation in real time. So we can, everyone can.
That monitor is having all the data, how much samples they have received, how much have been completed, how much have shown the errors, which one is supposed to be repeated, and those trays can be repeated for those samples. This monitor can control how much they have completed in that particular time. This is the central line, which I am telling the pulling of the samples. It itself takes the samples to particular instrument, either a coagulation laboratory instrument or the hematological autoanalyzers. And few quality persons are sitting over there. They were documenting all the time-based data of the instruments. And this is a TLA biochemistry laboratory. In this video you can see they do have a segregator also in this TLA solutions. One has to put all the samples which are having barcoding, just put all the samples in that instrument, and the instrument will recognize where it's supposed to go. This machine is a segregator. You just put the samples in this instrument.
For example, they will read the barcode of it is for immunology or chemistry, they separate. And also, if necessary, they make an aliquot sample by themselves.
So these, here you can see all the data which all these instruments are. All the instruments are connected with a single line. That's a single entry point where segregator, you put the samples, and it will decide at which instrument the sample is supposed to go. This is a clear-cut line. Here you can see. This one is a line. Through this line, the samples automatically goes to the particular instrument. All quality control. Here you can see that robot is waiting for the lift. It is controlled by Bluetooth. It operates the lifts, it can go take the elevators, it can go up and down, and it put the samples itself to that segregator also. Through these lines, these samples used to go. And the few areas like microscopy of slides is not possible. It do the centrifugation, but one has to see the slides of urinalysis. So one technocrat was sitting and she was analyzing the, the microscopy of those urine slides. This type of monitors used to, give the information of all the stats, like the quality control, how is it is running, whether it is in the limits, and how much samples they have processed. This laboratory used to process around 30,000 to 40,000 samples in 8 hours. So TLA helps only in this kind of laboratories where the samples quantity is very, very high. Here in the back you can see the pooling line. All these instruments are vertically connected through that line, and this itself comes, the racks used to come itself. These are the lines. Through this, these instruments are connected. All these monitor systems used to have all the data of that particular instruments. So at the time of having some errors, one has to go the root cause analysis. Once all the laboratories you will go for the accreditation, that error have been generated, but the root cause, who will do the root cause analysis? Our techno fellows are the key personals to identify these root cause analysis. We have to identify that which instrument have given the problem. So whatever the samples that will be processed through that instrument are going to give some problems. So we need to repeat all those instruments. Here is example of microbiology laboratory. In microbiology, we do have certain challenges. Majority of the work is used to be done manually. Only the streaking was automated over there, but we could not process. You can see all these plates are prepared by themselves. Streaking are available over there. But 2/3 of the samples which comes for the routine analysis, they used to be performed through TLA. As I said, that immunology and standalone testings, we need to have the automation, but we need to perform all the investigations manually only. Though there is automation, but one operator is supposed to process the samples, and the human touch is necessary in all these. You can see the HPLCs are there. These are the documentation, which is the most essential part of the accreditation. One has to manage all the documents, all the quality control testing reports and everything. Histopathology is as usual. It's manual totally. You can see the difference, which I can see like is more or less similar with our Indian scenario. So TLA can only be feasible for the high testing volume and the speed at which the samples been delivered through TLA. It also indicates that once there is a problem will arise, it can be rapidly multiplied and ultimately impact on the patient's reports. So therefore, qualified team of technologists along with the engineers, all these TLA instrument engineers and the IT personals who are going to manage all these data, because the errors, once we are going to analyze the root cause analysis, there are chances that certain software is not working, which is beyond our limitations. We could not manage that, so we need to have the IT fellows along with the team of the personals to manage all these facilities, and we all plays a critical role in this to recover the errors, reports. For the approvals of the reports, we need to have the human touch. Test validation, we require the human touch, and the programming of software also required qualified personals. So therefore, TLA provide helps as well as it is also very important that TLA laboratories need to have very qualified technologists. So some people used to think that once the TLA will be introduced, once the accreditation will be introduced, the importance of technologist is going to be reduced. I don't agree with that, because these instruments will help to deliver the quality reports with good precision and good quality controls. But for operating those instruments, along with to retrieve the root cause analysis, one has to have very qualified medical technologists. Those can identify the problems, because a minute problem if arise in a single lot, then thousands of the samples reports can be derailed. So our role will be there. Thank you very much.
Thank you, sir. That was a very lucid presentation. And also thanks for being spot on with the timing, because the organizing team wants it to be on time. So it was rightly said that instead of moving towards total laboratory automation, that it only augments more qualified technologies to be there, that human touch is always required for the success of automation as well. The session is open for any questions.
Good afternoon, sir. I am Ananya Gupta from Delhi Pharmaceutical Sciences and Research University. I'm a student. My question is, like, in the near future, there will be an advancement in the total laboratory automation. So what should we medical laboratory students should adapt to their skills so that we can remain relevant in the workforce in the near future? Thank you.
Being a student, I think you have to have control on your, the subjects which you are studying. So engineers, software fellows, they only can operate all those stuff, but they cannot identify. You can identify if a patient's SGOT, SGPT is elevated. You can identify probably their bilirubin bulk must also have been increased. So being a student, now for you, you need to enhance your subject knowledge. Basic knowledge of subject is very, very important. You need to have command on your all the subjects. Then once you will start doing practices, you will be having all the exposures, okay?
Hello. I have, though Dr. Sharma has given a very good this presentation, I could remember a time when Dr. Dinda, he was Dean Allied this All India Institute, he visited our PGI-MER. He was worried about himself. He was saying that very shortly pathologists will be no more, because so many scanners have come up, and these scanners can scan at least 300 slides in an hour. So what is the use of them to sit there? And these automations, these scanners are so sophisticated that they can diagnose also what disease is going to have, what type of cancer, it can classify also. So he was saying that pathologists will be no more. And today I think now technologists will also be no more once this artificial intelligence comes and this comes. Okay, qualified technologists are required, okay, to handle them. But I have to just say, as an academician, we have got this national education policy has given some provisions to have bridge courses. And certain courses you can opt for your own there are some courses that you can take as a optional subject, university called courses, in which you can opt for certain courses in which you have got inclination. You can opt for some engineering course or some computer course, means something which will hone you and your qualification, so that you will be in a position to fight with the future time to come with this artificial intelligence. This is just a piece of information to all of you. So you have to stand in front of such an advancement. Really, it was amazing to see these slides. Thank you.
Thank you, sir.
Thank you, sir, for the inputs. We promote science and we make sure that we also live up to it. With no further questions, I think, yeah. Yeah, here.
Very good afternoon, sir. You had a very good experience of TLA. This is Dr. Arti Tyagi from Gujarat, Charusat University. I would like to know, sir, you have visited the TLA laboratory. So what is their experience? Are they satisfied with this TLA, and are they lacking the shortage of manpower, of competent manpower skills?
Anyhow, this was a developed country and we are having sufficient workforce. We need to give the employment to our upcoming technocrats also. So TLA helps where the human hands are up. You know, once you had started your startup or laboratory and you are getting 20,000 of samples, how many medical technologists you will recruit to process in that small area? In those situations, you need to go for TLA. Dr. Lal Path Labs fellows, they must be having the TLAs, but our PGI, any of the government institutions, we are not having till date TLA. I don't know whether this new laboratory in the AIMS, they are maybe having the TLA, but as far as the human resource purpose in India, TLA is not feasible. In PGI itself, we have the standard laboratories, but we don't have the TLA solutions, because even though Department of Laboratory Medicine in India is not having the integration of microbiology, biochemistry, hematology, all these departments are separate, their specialist doctors are separate. So accordingly, I will endorse Sir statement. This TLA solution is going to take more and more jobs of medical doctors rather than the technocrats. So be rest assured, TLA is a solution only for the high number of samples.
So we can identify that for our students specially.
You will get the job.
I think this is a discussion which can be taken up later on. We are running short of time. We are already short up half an hour. So I would request you interact with the speaker later during the tea time. Thank you.
Thank you, sir. Requesting Dr. C.P. Pandey, sir, to kindly present the memento to sir. So please.
Our next talk will be Emerging Technologies in Medical Laboratory Sciences, presented by Ms. Anjana Kushwaha, RP Center, AIMS, New Delhi. Her introduction will be done by Dr. C.P. Pandey.
Well, welcome. With a huge round of applause, we welcome Ms. Anjana Kushwaha. She is a Senior Technical Officer at Dr. RP Center for Ophthalmic Sciences, AIMS, New Delhi. She has completed her Bachelor of Education and Master degree in Plant Science from Lucknow University. She has more than 24 years of experience in laboratory and is trained at AIMS, New Delhi. She has knowledge of basic biochemistry techniques, including bioanalyzer, immunoassays, cell culture, and flow cytometry. So over to you, ma'am. And we have already overshot our time, so I would request stick to time. We'll rather be encouraged if you finish off lesser than your time, right? Thank you.
Thank you, sir. And good afternoon to my seniors, my colleagues, and our future technologists. Welcome to AIMS New Delhi ICMLS-2024. My topic, Emerging Technologies in Medical Lab Sciences. Medical sciences is the field that is continuously developing. It is very necessary for the future pathology and the diagnostics to keep finding and achieving new milestones. It gives us the tools to look at how our body works in depth and allows us to translate that to the clinical fields. In terms of a lot of the diseases that are happening, especially with the recent pandemic COVID-19, the most used test, RT-PCR, it really places spotlight on the significance of studies of pathology and the emerging technologies. The new technologies and the skilled technologists will place pathology in the forefront of diagnostics by producing precise and accurate reports so that the clinicians can diagnose and treat the patients in the best possible way. Today, the massive emphasis is being given to skilled, innovative technologists who want to learn new techniques and decipher the hidden mysteries in human body. Evolution in hematology laboratory technology for differential cell counting. A little bit history of evolution. The WBC counts were usually performed manually using the hemocytometer, and today refining the automatic differential by introduction of IG count into CBC, immature granulocytes count in CBC. Then knowing about evolution in immunology, how immunotherapy progresses through the ages. In 17th century, variolation against smallpox replaced by vaccination with cowpox by Jenner. In 1950, discovery of cancer-specific antigens, body irradiation and bone marrow transplantation for treating leukemia. In 1980, Nobel Prize Serotherapy against diphtheria by Behring. 2018, Nobel Prize Cancer Therapy by Immune Checkpoint Blockade, Ellison and Honjo. In 2020 is Nobel Prize for CRISPR-Cas, Genome Edition, Charpentier and Doudna. They engineered immune cells of multiple lineages, including B cells. CRISPR is the latest technology that allows researchers to modify the DNA of living organisms. It was discovered in microbes like bacteria, archaebacteria. As we move from the earlier to the latest technologies, flow cytometry is a lab test that uses a laser to detect and analyze the physical and chemical differences of cells and particles. As you all know, it's a powerful technique that can be used to diagnose conditions like cancer and infectious diseases. Flow cytometry works as cells are labeled with fluorescent components. Passage cells pass in single file in front of a laser. The laser excites the fluorescent components, causing them to emit at different wavelengths. Detectors capture the light that emerges from each cell. Software analyzes the data to report on cellular characteristics like size, complexity, phenotype, and the health. Advancement in molecular diagnostics, that is polymerase chain reaction, PCR, as you all must be doing it also and must have learnt also. PCR is widely used technique for amplifying specific DNA sequences, enabling the detection of pathogens and genetic mutations. Real-time polymerase chain reaction, a higher version, and then the microarrays. Microarrays allow for the simultaneous analysis of thousands of genes or proteins, providing comprehensive insights into cellular function and disease states. dPCR, digital PCR, is based on the principle of endpoint PCR, but the PCR reaction mix is split into thousands of single partitions. The nucleic acid template is randomly distributed across all available separations and amplified individually. Sanger sequencing, then next generation sequencing. NGS technology allows for rapid and accurate sequencing of DNA and RNA, providing detailed genetic information for diagnosis and personalized medicine. Polymerase chain reaction, which is widely used, has limitations, poor precision, low resolution, low sensitivity, etc., which is it is not quantitative test also. Then we do the real-time polymerase chain reaction. It monitors the amplification of a targeted DNA molecule during the PCR, not at its end as in conventional PCR. The advantages of real-time PCR are post-PCR processing not required, it is quantitative, it is precise and accurate. Microarray is a laboratory tool used to detect the expression of thousands of genes at once or to identify specific genetic variations. Principle of DNA microarray technology is based on the fact that complementary sequences of DNA can be used to hybridize immobilized DNA molecules. There are 4 major steps in performing a typical microarray experiment: sample preparation and labeling, hybridization, washing, and image acquisition and data analysis. Now, digital polymerase chain reaction is a revolutionary technique to precisely quantify nucleic acids for its high sensitivity and specificity. dPCR is a type of PCR approach that partitions a sample into thousands of individual compartments, allowing for absolute quantification of target nucleic acids. dPCR involves partitioning the PCR solution into tens of thousands of nanoliter sized droplets, where a separate PCR reaction takes place in each one of the cell. Advantages of digital PCR: no need to rely on references or standards, which was said earlier or seen earlier. Desired precision can be achieved by increasing total number of PCR replicates, more tolerant to some PCR inhibitors, provides a linear response to the number of copies present to allow for small fold change differences to be detected. Sanger sequencing, which is being done in IRCH, principle of chain termination, where during the PCR growing DNA strand is terminated by the incorporation of di-deoxynucleotides, A, T, G, and C in different reactions. The fragments of different lengths of the amplicon are then run on a gel in different lanes to determine the sequence. Next generation sequencing. Next generation sequencing is a technology for determining the sequence of DNA or RNA to study genetic variation associated with diseases or other biological phenomena. Again, it is being done in IRCH in AIIMS, New Delhi. 2 major paradigms in NGS technology. Short read sequencing provide lower cost, higher accuracy data that are very useful for population level research and clinical variant discovery. Long read approaches provide read lengths that are well suited for de novo genome assembly applications and full length isoform sequencing. Sample process is done like this: sample is taken, then library preparation, sequencing, and bioinformatic analysis. For NGS, the bioinformatic analysis is the major part where still people are lacking. That is what it is. There is a need of standardized protocols and robust bioinformatics infrastructure. Robotics and automation in lab workflows. Automated sample handling, robots can handle samples with precision and speed, reducing manual labor and minimizing the risk of errors. High throughput screening automated systems can analyze thousands of samples simultaneously, accelerating drug discovery and research efforts. Reduced turnaround time. Automation leads to, that is, automatic machines which can handle and do everything on their own without the need of a human doing it. So automation leads to faster results, enabling quicker diagnosis and treatment, which benefits patients and improves workflow. Artificial intelligence and machine learning applications. Image analysis. The artificial intelligence-powered algorithms can analyze images from microscopy and other imaging techniques, assisting in the detection and diagnosis of diseases. It can do in a more speedy way. Point of care testing and decentralized diagnostics. Improved patient experience and faster results, and it can reach the remote places where human cannot go. Lab on a chip and microfluidics technologies. This is done in a chip, a small chip. The test can be done, the whole test, which we do in tubes and the other systems. Lab on a chip devices integrate multiple laboratory functions onto a single chip, reducing the need for bulky equipments. Point of care testing and decentralized diagnostics. Wearable and implantable biosensors. Now there are machines which we can wear, a patient can wear, and throughout the day, and the data of the whole day can be fed on it, or it can be there. 2 minutes more, sir. So cardiac monitoring, wearable sensors can monitor heart rate, rhythm, and blood pressure, etc. Integration of lab data for electronic health records. Improved patient care, reduced errors, enhanced research. Ethical considerations and regulatory challenges are there. Data privacy and security has to be kept. Artificial intelligence and automation in healthcare. Ensuring maintaining human oversight and accountability is the paramount. Thank you. Hope we need more technologists who are skilled in IT and who have the knowledge of bioinformatics, who can deal with the data which we have collected so much, and who can, in NGS and all the techniques, they can decipher what the data is telling us. Thank you.
Well, it was a wonderful presentation, ma'am. Thank you. A quick question, if it's there. Otherwise, we are 30 minutes behind our schedule. Any questions? So, thank you so much. Maybe your talk was so informative and clear that there is no question as such. Thank you, ma'am. Thank you so much.
It was an informative talk only. All must be learning the techniques, and still there is so much of scope that they can learn more. Everything is available, but the thing is, we need more skilled technologists. So you have to study more.
Right. Thank you. Thank you.
Our next talk for the afternoon is from Petri plate to sequencing, recent advances in medical microbiology diagnostics, presented by Dr. Monica Mahajan from Postgraduate Institute of Medical Education and Research, Chandigarh.
Hello. You forgot to honor her.
Sir?
Forgot to honor her. Honor her.
Honor. No, sir. Valedictory. Dr. She'll be doing Dr. Rathor's intro. She's saying that I'm introducing before. Welcome Dr. Thirumurugan to introduce Dr. Monica Mahajan.
Thank you. She works as a quality manager and technical assistant in Department of Microbiology, PGI, Chandigarh. She has a lot of publications, both national and international. She's a reviewer of emerging infectious disease, a high impact factor international journal, recipient of travel grants and many prizes in CMAs and conferences, chairperson of AHP Con and ICMLS conferences. She has delivered talks in AHP Con and ICMLS conferences. Her paper has been selected for the award of oral presentation in ICMLS. And there's a lot more to continue. For shortage of time, ma'am, it's up to you.
At the very onset, I would like to thank the organizers for giving me this opportunity to present on this prestigious platform. And thank you, chairpersons, for the nice introduction. So today, I'll be talking on from Petri plate to sequencing, changing diagnostic paradigms of medical microbiology.
Sorry for interrupting the beginning itself. We would like to have it concise for 10 minutes for shortage of.
Yeah, I would rather try.
Thank you.
And thank you, Dr. Surendra and Ms. Anjana, for setting a platform. I'll be only touching the microbiological aspects. So before I start, let us remember founders of microbiology. Everybody of us talk about Robert Koch, Louis Pasteur, but one of the unsung hero is Julius Patry, who has given us Patry plate, and because of him only, the diagnostic conventional microbiology is possible. Then another heroes are Watson and Crick and Kerry Mullis. We all know about him. Watson and Crick gave us double helix structure of DNA, and the Kerry B. Mullis and Michael Smith, they were the Nobel Prize winners in chemistry in 1993 for giving us PCR. So three more heroes who are Frederick Sanger, along with his colleagues Ellen Max and Walton Gilbert, they gave us sequencing platform. Ms. Anjana has already discussed it. So another landmark paper which showed CRISPR-Cas9 as gene editing tool, it was published by Emily Charpentier and Jennifer A. Jordana. So now if we come to diagnostic microbiology, it focuses on identifying pathogens and immune responses responsible for infections. The purpose is to inform appropriate treatment and understand disease infectious patterns. So the importance is that rapid and accurate diagnosis should be there, which is critical for effective healthcare. This in turn reduces turnaround time. The traditional diagnostic techniques, as all of you know, are microscopy and culture techniques, but there are some drawbacks of the conventional techniques, like they are time consuming, require skilled personnel, limited sensitivity, pathogens in viable but cultural, non-culturable state cannot be detected, delay in appropriate treatment, and they are more labor intensive and needs more resources. So, many advances have already been talked about to previous speakers. So directly I will come on microbiological perspective. Sanger sequencing has already been talked about in detail. It is a high throughput sequencing that identifies pathogen from genetic material. The applications are in metagenomics, outbreak tracking, and finding antimicrobial resistance genes. These are comprehensive, but they are expensive and require data analysis expertise. So, some of the more advancements in medical microbiology diagnostics are real-time PCR, gene expert, and other modifications. As we all know, real-time PCR can quantify pathogen load. Multiplex PCR assays, which are used in diagnosis of respiratory, gastrointestinal, and sexually transmitted disease with the syndromic approach. Nowadays, the kits are available which go for syndromic approaches. This helps clinicians in giving a targeted therapy. One of the very important tests is Gene Expert and PreNet. I will be talking about this little bit in detail in my subsequent slides. Hotstart PCR, Touchdown PCR, and digital PCR has improved sensitivity and specificity. LAMP, it has made diagnosis of tuberculosis easy even in field. It is a WHO endorsed test and RNTPC recommended test. So let us see difference between what is difference between Gene Expert and Gene Expert Ultra. These both tests are used for diagnosis of MTB complex, but in MTB RAF we use real-time PCR, and in Ultra nested PCR is used, so it makes the test more specific. And also the detection limit. Here the detection limit is 114 CFU per ml, and in Ultra detection limit is 16 CFU per ml. These two are very promising tests for diagnosis of TB and rifampicin resistance. So now what is TrueNet? TrueNet is a chip-based RT-PCR that detects, initially it was used to detect tuberculosis. Later on, during the COVID era, it was used to detect SARS-CoV-2 gene, which is responsible for COVID-19 infection. It is first kit in India to receive emergency use authorization by the Drug Controller General of India for diagnosis Nipah viruses. So now I come to CRISPR-Cas, which is Clustered Regulatory Interspace Short Palindromic CRISPR associated. Ms. Anjana has already elaborated on this. So briefly I will talk about that CRISPR system has been divided into 3 subtypes, type 1, type 2 and type 3. And in this discovery was made by Emily Charpentier, I have already told. So in microbiology, it is used for genome editing. It is a genome editing tool, and it is also used for detecting antimicrobial resistance. In infectious diseases, it can be used to detect COVID-19 and malaria, and it is also used for understanding host pathogen interaction. And recently, CRISPR-Cas 13-based system are used for antiviral therapy. So now I will come to some of the advancements in immunological techniques. As we all know that flow cytometry can be used to analyze specific cells in samples. They are used in immunophenotyping. So recent developments are multiplex immunoassays for simultaneous detection of multiple pathogens and chemiluminescence immunoassays, and one of the advancement is mini-RADAS, which detects Clostridium difficile toxin A and toxin B. Now if we talk about the advancements in diagnostic mycology, beta-D-glucan and galactomannan are used. So also we are doing therapeutic drug monitoring in case of detecting the harmful effects of antifungus by HPLC. So this is very important slide, especially for students. 2 important tests, beta-D-glucan and aspergillus galactomannan are widely popular amongst clinicians. So they detect antigens for Candida and aspergillus, and galactomannan will detect antigen for aspergillus. But the exceptions are that beta-D-glucan is negative in Cryptococcus mucorales and blastomyces, and aspergillus galactomannan is negative in mucorales, because the cell wall does not contain these two. And also there are many false positives. It has to be interpreted with caution, as if the patient is on intravenous penicillin and various other factors are there, beta-D-glucan will be positive. And if the patient is on piptase, amoxiclav, then aspergillus galactomannan can be positive, and many other false positive scenarios are there. So let us talk about lateral flow assays. Many lateral flow assays are available for microbiology diagnostic. Like one is Typhi-DOT for typhoid, Cryptola for diagnosis of cryptococcosis, NPT64 antigen for detection of MTB complex. For lateral flow assays are also available for Plasmodium vivax and falciparum, and also for dengue. Now, point of care test. As the definition suggests, the diagnostic test which can be performed
near the patient, they are the point of care tests. Some of these lateral flow assays can also be used as point of care tests. Examples are COVID-19 rapid tests available for diagnosis of influenza and for malaria, for dengue, for cryptococcus, and they are very quick, convenient. So, some of the diagnostic tests which are available for virology in our institute, they are dengue serology. Like multiplex PCRs are available for all the 4 serotypes of dengue virus. PCRs are also available for adenovirus, dengue virus, chikungunya, Zika virus, varicella zoster virus, enterovirus, and also multiplex for respiratory viruses are available. And quantitative PCR are being done for hepatitis B, hepatitis C virus, and cytomegalovirus. So, molecular parasitology is not far behind. So, PCRs are also available for diagnosis of amoebiasis, giardiasis, hepatitis, acanthamoeba infection, toxoplasmosis, echinococcosis. But lab is not standardized till now. It is still in infancy. And also, AI is in microscopy is available to detect Plasmodium species. So now I come to automated methods. Many automated methods, all of us might here know that there are BACTEC, BacTalert, and many more. For microbacterial growth index tube. For urine microscopy also automated methods, automated microscopy is available, and even for urine culture they are available. By nepherometry we can detect anticephalocin O and CRP, Vitek MS which processes sample more efficiently and quicker. Gives quicker antimicrobial susceptibility testing with higher accuracy is available. MILDITOF MS, all of us know, which provides rapid identification of bacteria and fungi. So this is MILDITOF, Bruker's MILDITOF machine. MILDITOF provides rapid identification based on the unique protein signature. It really has caused paradigm shift in diagnostic microbiology, as the results are available within few minutes. So it is based on protein fingerprinting. The benefits are it is fast, accurate identification, but the drawbacks, especially students to note, it cannot differentiate amongst Salmonella species. It identifies Shigella as E. coli. So again, the conventional diagnostic methods will help in differentiating species here. So lastly, I will talk about in brief about the Human Microbiome Project. It is a National Institute of Health research project which studied human microbiome, which emphasized on the role of commensals in health and disease. Sequencing technologies and bioinformatics have enabled the characterization of microbial communities, which help clinicians understanding the role of dysbiosis in inflammatory bowel disease, obesity, and infections. So, as we all know that distribution of microbiota, different sites of bodies have different microbiome species. So there are very interesting facts about human microbiota. There are 10 times as many microbial cells in the human body as compared to human cells. The number of genes are also 100 times. It is estimated that 20 lakh genes are there in bacterial species and only 20,000 are there in human genome. And the microbiome is essential for human development, immunity, and nutrition. And also it has been seen that the microbiome of the mother may affect the health of her children. With this background information, the Human Microbiome Project, which was started by National Institute of Health, NIH, was established in 2007. The project had 2 phases. Phase 1, which was from 2007 to 14, which had a goal to develop a reference set of microbial genome sequences and to perform preliminary characterization of human microbiome. Then there was phase 2 from 2014 to 2016, which was known as Integrative Human Microbiome Project, and the purpose of these project was to use omics technologies. So various omics technologies were used in metabolomics, lipidomics, and immunoproteomics. So the major three components were diabetics and role of human microbiome in inflammatory bowel disease and also in preterm birth. So the human presence of specific human microbiome was related to all of these. So now coming to AI in diagnostic, already been mentioned by previous speakers about robotics and total lab automation. It enhances accuracy, process large data, and the pathogen identification is easier. Drug susceptibility predictions can be there, and it can be used for reverse vaccinology. Last but not the least is lab on a chip miniature devices, which has already been mentioned by Ms. Anjana. So there are some challenges in automation. The cost is very high, need of skilled personnel is there. So ensuring accuracy, handling patient data is very important. The take-home message of my presentation is continuous innovation and advancements are transforming infectious diseases. AI and automation cannot substitute clinical reasoning in medical microbiology, which this AI cannot comment on whether the organism isolated is a normal flora or a commensal or a pathogen. It cannot judicially tell about significant bacteriuria without knowing clinical history, where even low numbers are important. Contamination and infection cannot be differentiated. And lastly, we are a resource limited country, so this automation is not possible in our country. The future directions are CRISPR-based technologies and biosensors, they can be really developed, improving accessibility to develop cost-effective portable devices. Thank you all for your advertent attention.
Thank you, ma'am. That was a very intensive and informative presentation. We completely acknowledge that that's a very vast topic to be concise into 10 minutes or a couple of minutes more than that. But we think that you did a complete justice to your talk. Considering the constraints of time, all the questions will be taken at the end of the session, or it can be put directly to the speaker. Okay. Thank you.
Thank you, sir.
Thank you, ma'am. I now invite Dr. Ruchi Rathore, Associate Professor of Department of Pathology, AIIMS, New Delhi, to talk about the role of medical laboratory technologist in the era of automation in histopathology. Welcome, ma'am. I welcome Dr. Malliga to introduce her.
Welcome, ma'am. And the special session speaker, Dr. Ruchi Rathore. She is also from AIIMS, Professor, Department of Pathology, and she is specialized in breast pathology and gynecological pathology. And she co-authored one chapter in well-known book Robbins and Cotran, and she has many publications to her credit. And currently she is working in the research project novel molecular subtypes of uterine sarcoma. Thank you.
Thank you. Thank you very much, ma'am. So I thank the organizers for this opportunity and the chairpersons for this kind introduction. Without taking much time, I would like to start with this presentation. So basically, we have discussed that we are going towards automation in hematology, in microbiology, and so in histopathology. So I'm giving a brief overview of what are we as lab technologists supposed to do with automation in histopathology. So the role of medical lab technologist in the era of automation in histopathology. We all know that before that the traditional histopathology laboratories have included receiving of the specimen, grossing them, and then manually processing, embedding, sectioning by microtomes. The staining was done manually. After the diagnosis, even the IHCs for diagnosis were done manually so far. But now with automation, there is, with automation in histopathology at literally every step, we see that the tissue processing is done with the help of automated tissue processors to embedding, to microtomy. Even auto stainers are being there for giving us automated automated staining of the slides, giving high quality slides. For IHCs, we have immunostainers these days, and the quality of the slide actually tells us and helps us in scanning these slides. So at every aspect, automation has entered in histopathology. With this automation, it provides good precision, it reduces the turnaround time, it reduces human errors, it improves the consistency. And also it is able to handle very high volume of samples, especially at centers like ours, where we cater to thousands of samples on a routine basis. This has streamlined many routine processes. Apart from this, automation has given freedom from repetitive tasks to our technologists, but it doesn't mean that the role of technologists ends with automation. In today's era, the technologists, their expertise, vigilance and adaptability remains central to ensuring the accuracy and the efficiency. The machines will be working, but it is the role of our modern day technologists to give us efficient and good results with these automations. Okay, so the evolving role of lab technologists vis-a-vis earlier, where there was the functioning involved routine processing and routine embedding of the tissues. This shift has been from routine to specialized tasks. In today's era, the lab technologist is only not limited to cutting of the sections, but they have to be specialized in quality assurance, in overseeing the functions of the machine, and they have to know the complex troubleshootings of every machine they are dealing with, be it tissue processors, be it auto stainers, be it Ventana systems of IHC. They have to develop their skills to acquire, basically they're expected to acquire new skills to operate, maintain and troubleshoot these automated systems in histopathology labs, so that even digital pathology and AI integrated tools can be handled with the technologies of today's era. So we have to put this in mind that automation is not replacing but transforming the role of technologists today. It is now that technologists are now expected to supervise and ensure the accuracy of these automated processes. So coming on to the areas where automation has entered the histopathology labs, it has started from sample processing and staining to quality control and digital pathology and image analysis. Now when machines are doing the work, they are doing some amount of quality control, and we may think that quality control with machines is an easier task, but it is not true. The point is that with automation, the role of technologists enhances and the importance of quality control increases. The technologists today are responsible for regular calibration of these machines, conducting regular reagent checks. Cleaning of these machines is very important for the proper functioning and maintaining an error-free environment. Now with the automation, there are challenges that come up, and our technologists today should be well versed to know what are the challenges that could come up and how to handle them. The first and foremost includes the technical challenges. Right from operation of the machines, these machines can break down anytime, they can have calibration issues, they can have software bugs. So the technologists today who are involved in histopathology labs must be proficient to resolve these issues very quickly. Because automation also gives a large amount of data, there are huge amounts of data involved, so the role of technologists is not only limited to the lab, but they are also responsible for maintaining these large volumes of data and to oversee the functioning of these automated systems. Apart from, so these automated systems in histopathology labs, apart from reducing the manual strain and faster turnaround time, it also gives an opportunity for the younger generation of technologists to basically specialize in other areas like machine management, quality control, and data analysis. Now with automation, basically the responsibilities of the contemporary histopathology technologies have completely redefined, and it goes into every area. Starting from sample management, in earlier eras there used to be registers for everything, but today they're in the pre-analytical stage, proper collection, labeling, fixation and processing of the samples is extremely important for their integration with the automated systems in the labs. Moreover, in the labs where the technologists are involved with grossing of the sections, it is important to currently orient and trim the sections so that they can be identified by the systems properly. And we have digital archival systems for storing the slides properly. So this is the image from our lab which shows the LIS integration. Our LIS has been integrated very wonderfully with the automated labs that we have, and our technologists, I'm very happy to say that our technologists have upskilled themselves and they're doing very great job in actually taking such loads of samples today. This is our new slide archival system which we have just installed. It is functional now. Vis-a-vis earlier systems, it is now the system is automated, it is integrated with the LIS, and slides and taking out slides from the archival system is much more simpler as compared to earlier. Now the most important part comes in operation and maintenance of the automated systems. As we all know that machine setup and operation requires a skill of the technologist. So preparing these automated systems with correct reagents, programming protocols and monitoring workflow is very important. Look, every histopathology lab has a different load, and as per the load, it is the role of the technologist to set up these programming protocols and to monitor the workflow of the machines. In troubleshooting, it is very important for diagnosing and resolving the technical issues. Quite frequently, the technical issues sometimes comes up with the machines, and in those cases, it is essential for the technologist to understand where is the problem and how can it be resolved as soon as possible. Then, the regular routine checks and cleaning and calibrations are extremely essential for working of these automated systems, because these machines are so sensitive that even a small amount of wax can stop the tissue processor and the whole day of work can be wasted. So it is the role of the technologist to be vigilant in every step and to oversee these machines and their functioning. Quality control and assurance, they have to oversee the routine maintenance and calibration of these systems. Now, validation of result is something very important for a technologist. Why? Because when the auto stainer is giving you a slide, it is often, sometimes it is overstained, sometimes it is understained, and if the test run, so it is important for the technologist to basically run a test run before every system and take care of the machines so that the whole lot is not wasted. So validating the quality of the tissue sections, the staining and the digital images to ensure good diagnostic output is of utmost importance for the technologist. Error prevention is also an important role of technologists, where how to identify and rectify these errors or inconsistencies in automated workflows. Commonly it involves reagent issues or machine malfunctions. There will be company guys who will be helping in these, but at first step the technologist should be able to identify these steps and be able to rectify this if as far as possible. These are our automated systems in the lab. This is our newer tissue processing systems. These are fully automated. This is a semi-automated system. This machine, as you see, it shows all the levels of reagent, what needs to be done, but it is the role of a diligent technologist to basically ensure routine functioning of these. It is very important to clean these machines. The daily and weekly cleaning tasks are important. You have to see that the reagent bottles are fine and the used wax is discarded. So, and the touch screens are cleaned properly, the filters are changed regularly. All these things have to be handled by the skilled technologies today in our labs. This is our auto stainer which is installed in our lab and has been functioning very nicely with the help of our skilled technicians and our skilled lab. So they make sure that before every run the machine is cleaned properly, the devices are clean, and the reagents are removed, and a test run is done on daily basis before running of the lot for the day. So now coming on to these are the machines. This is how a tissue processor shows. The machine has its own quality control manuals and the checks, but it is the responsibility of the technologist to oversee where they are going wrong. Like in this image, it is showing that alcohol needs to be replaced. So it will show, but on daily basis you have to check it and to see what needs to be replaced. Similarly, for tissue stainers, you have to check that all the reagents are fine, hematoxylin is of good quality, so that whatever slide you get is of good quality, the staining is of good quality and of reportable quality. You have to be well versed with the troubleshooting steps. Now, apart from knowledge of how to cut, of the section of the thickness, how much to cut, how is the slide, it is now an added thing to know these troubleshooting steps for the machines these days. Like basic steps, like sometimes the machine can show errors like these, where you have to ensure that the timer of the machine is set properly. Then there are some system errors or changing filter errors. These all can come, which you have to be aware and not panic when the machine stops working. Right. Sometimes the machine can show basket error, where you have to be aware that how to basically correct this, if the basket is placed properly or not, if the slides are placed properly in the basket or not. Also in today's era, apart from the rapid turnaround time, the technologists also provide support for high throughput workflows. In high volume labs like ours, the technologists are vital in coordinating automated processes to handle multiple cases efficiently. Now, today's modern day technologists are not only limited to just labs, they are involved in a lot more things. So collaboration is a key skill for the newer, younger generations of the technologists who are still developing and training themselves. Apart from working with the pathologists in preparing high quality slides, or addressing the queries related to processing, or providing technical input when needed, they have to be in liaison with the IT and the bioinformatics guy. So why? Because every lab or every hospital today is integrated with the LIS system. So how the LIS system works with automation, technologists has to be well versed with that as well. Moreover, in research with the techniques like advancement in techniques like IHC and FISH, molecular testing, our technologist has to be well versed with these things, with these techniques also. So this is our automated immunostainer, which stains the IHCs automatically. But I would like to tell and congratulate our IHC staff also, because up till now we have an IHC load of 300 or 400 IHCs on a daily basis. They were handling it manually, but they have very nicely and smoothly transitioned to a automated system with so much of load, and they are able to troubleshoot where the staining IHCs are going wrong or going right. And today we are having a smooth functioning with even 400 to 500 IHCs per day, thanks to the technical skills of our technologist. So I would, just the last two slides. I would like to say that for a younger generation of technologists, basic knowledge of your medical laboratory books is important, but apart from that, you have to upskill yourself and be well versed with the automated systems, with the software, how do they work, with the techniques of these automated systems, and you have to train your younger generations in the same. Also, you have to be sticking to adhering to the accreditation standards, because all the labs are accredited these days, and data security, I think. This is the last slide, digital pathology. If the quality of the slide is good, you can go ahead with telepathology, and digital pathology slides can be scanned properly. So in that also, you have a very essential role to play. This is our digital slide scanner, which can scan 360 slides at a go. And now what the future holds for the younger technologists today is that how to basically steer this era of automation is by training and upskilling yourself. There is a need for ongoing education to keep up with rapid advancements, and collaboration is a core skill, and you have to adhere to the ethical guidelines and accountability of the patient reports. So this is my final slide, your take-home message, that automation enhances and rather than replaces the role of lab technologist. Today, the role of medical lab technologist has transitioned from purely manual tasks to a more sophisticated, technology-driven role that requires a blend of traditional skills and new technical competencies. Your expertise ensures that automation enhances the standards required in histopathology labs. With this, I thank the institute, my department, and my lab staff who have worked very hard to give us very good results. Thank you very much. Thank you, sir.
Well explained each and every skills and technique in histopathology laboratory.
Thank you.
Thank you.
Thank you. Thank you, everyone.
Thank you, ma'am, for the interesting talk. We now request Dr. Thirumurugan and the rest of the chairpersons to honor our speaker with a memento. Pankaj, Tirumuruga. We also request Mr. Fakhruddin to join the chairpersons to give a memento to Ms. Anjana Kushwaha. Thank you, sir. Thank you, ma'am. So the next plenary session is on quality, patient safety and career pathway. Chairpersons for this will be Dr. Malliga, Dr. Pramod Kumar Verma, and I request Mr. G.S. Vadavan, President JFMTI, and Mr. Ajit Singh to join the chairs. Dr. Malliga is a resource person in many conferences and presented scientific papers. She has organized many programs for celebration of NMLPW, CMLE, and other professional events of MLS. Dr. Pramod Kumar Verma, he is a medical laboratory technologist in molecular biology lab in Dr. Bhim Rao Ambedkar Institute, Rotary Cancer Hospital, AIIMS, Delhi. He is working on leukemia for molecular diagnosis and monitoring. He holds PhD and postgraduate diploma in medical laboratory technology from IMS BHU, Varanasi. He has published 4 international and 7 national papers. The next talk is on transition to new ISO 15189-2022, major changes and impact by Dr. Malkit Singh, Dean Dolphin PG College of Life Sciences. I request Dr. Pramod Kumar Verma to introduce the speaker. I request Mr. Ajit Singh to please come on stage to join the chairs.
नहीं ठीक है कौन सा दूसरा इसका वो कहाँ है पॉइंटर ये तो नहीं इज इट स्लाइड्स आगे पीछे
Welcome Dr. Malkit Singh ji. Dr. Malkit Singh needs no introduction, I think. He is very much senior experts from the laboratory sciences and he is retired, but he is continuing putting his efforts in encouraging his academics and making more inspirations to the youngsters. You are welcome to give your speech and lecture, please. Thank you.
Thank you, chairpersons. And good evening, everybody. The topic which has been given to me is the transition to new ISO 15189-2022, major changes and the impacts. ISO 15189-2020 is an internationally recognized standard that specifies requirements for quality and competence in medical laboratories. It reflects the need for a harmonized approach to ensure reliable and accurate medical laboratory results, which are essential for patient care and safety. ISO 15189 was developed by a group nominated by the ISO 17025 team. There are two standards there were before 2003. The standards were ISO 9001, which was in general quality management system, and along with this, the new standard developed was ISO 17025, which was general requirement for the competence of testing and calibration laboratories. The need for a new standard for medical laboratories forced the ISO 17025 group to develop some standard for medical laboratory testing. Hence, that group developed ISO 15189, which was driven by growing demands for quality assurance in healthcare. It drew upon the two existing standards. Those were ISO 9001 and ISO 17025. These standards provided the basis for a specialized standard tailored to the unique needs of medical laboratories. So this line also shows that previously the standards were there for general laboratories, but there were no standards for clinical laboratories. And the first edition of ISO 15189 was published in 2003. It emphasized the quality management system and technical competence in medical laboratories. And it was tried to make in such a way that it should bridge the gap between quality management principles and the specialized requirements for clinical diagnostics. After that, a few more additions or revisions were done, and these undergone many, the standard undergone many revisions to keep pace with the advancements in technology, in regulatory expectations, and best practices in the healthcare. It was again from 2002 to 2003. In 2007, it was again revised and introduced more in a more detailed guidance for laboratory accreditation, which was placed with the greater emphasis on risk management and continual improvement. Then after 2007, it was revised in 2012, which was focused on aligning the ISO 17025 and ISO 9001 revisions. Actually, at that time, what was happening, some laboratories were having ISO 9001 as general standard, general quality management standards. Some were having ISO 17025, that was for testing any of the testing laboratories, but there were no standards of ISO 15189. Now when ISO 15189 came, then problem occurred that the laboratories which are already having ISO 9001 and 17025, they had to keep another third standard. So documentation was a lot. Then a desire or a requirement was generated to combine these three. So now the system is going on that in the clinical laboratories, there should be only one standard, which should include ISO 17025 standards, and 9001 standards also. So 2022 was introduced with several significant updates compared to ISO 15189-2012 version, reflecting advances in medical laboratory practices and an increased emphasis on risk management and quality systems. Now, when you will, I think from January 1st, 2025, there is a mandatory that you will go for only ISO 15189-2022. You won't be able to get 2012 now, accreditation certificate. So, one major thing came up and some people asked this question. I have not even included in my PPT, but many people asked, usually because in my area I am an internal auditor as per NABL. So people ask that, some people say that we don't need quality manual now. Do we? See, now this particular standard, since this has been made in combination to ISO 17025 and 9001, the authorities say that manual is not required, but it is not mandatory that you won't make the manual. You can make the quality manual. Nobody will stop you. That will make your documentation easy, maintaining of documents easy if you go for quality manual. But no assessor will ask you, or no assessor will put in non-conformity if you don't have quality manual. Now we'll discuss some key differences between ISO 15189-2012 and 2022. Very first thing is the structure alignment with ISO 17025 and ISO 9001. We have already discussed the revision was made to align more closely with ISO 17025 so that no three different quality manuals are to be kept. Only in 15189, almost everything which is required in 170, which is already present in 17025 and 9001, that is included. Another thing which has added new, that is a risk-based thinking. A strong emphasis has been given on risk-based thinking in ISO 15189-2022. Laboratories are now required to adopt a proactive approach to identify, evaluate, and mitigate risk associated with all aspects of the laboratory work, from just starting from sample collection, that is pre-analytical, from pre-analytical to then post-analytical, analytical, post-analytical, on all the processes. Risk-based requirements are there, where you have to assess the risk, you have to go for risk assessment, then risk management. If such a risk will occur, if you have identified that this may occur, then before processing, before reaching the processing, before sample collection, before processing the samples, you will tackle that situation that this type of incidents will not occur. So, first of all, the major document which is being asked by auditors these days, that is the risk management document, where they ask for risk assessment and the precautions to be taken to mitigate those risks. Now this extends to pre-examination, examination, post-examination processes. Now impact of risk-based thinking in ISO is there. The inclusion of risk-based thinking in the version reflects a modern approach to management system, emphasizing proactive identification and mitigation of risk along with the opportunities, not after the incidents happens. The competence requirement, another is, it was also present in 2012, but there a lot of stress was not being given. Now, in this 2022, more focus is given on ensuring that personnel should have the skill, education and training to perform their task competently. Their qualifications, their experiences are being now counted. Another thing which has been added to this is the inclusion, the point of care testing. Now previously, point of care testing was not included in 2012 edition, but in 2022 edition, the point of care testing has also been added. This is added to align the routine tests which are being done in the laboratory to the point of care testing tests. The quality management system integration. This I have already told you that quality management system as per 9001 and 17025 has been integrated in 2022 edition. It emphasizes the need for a robust quality management system that ensures consistent quality across all the activities. Another thing is information management. Up to 2012, everything was required at least on the registers. But now a stress has been given to the laboratory information system or hospital information systems, online systems, electronic systems. Now they say that you just discard your registers, go to online. But still, those people who will be in transition period, they will be allowed to keep the records on the registers. Then continual improvement and corrective actions. Continual improvement, you will take feedbacks, you will entertain the complaints from feedbacks, complaints and staff suggestions. You develop a criteria for continual improvement in your work. And in 22, a lot of stress has been given on corrective actions. Previously, what happened if some incidences happen? We might be doing corrective actions, but we were not giving in written. We were not taking it onto the record. But here, after 1st January 25, you will have to keep every record of the incidences and the corrective and preventive actions which you will take. Updated terminology. Some terms have been updated. The terminology used in ISO 15189-2022 has been updated to reflect current industry practices and the language of the related standard, especially ISO 17025 and ISO 9001. Means the languages also, the language, I don't mean English style of the language written in 17025, 9001 and 15189 are now same. For example, there is more comprehensive use of the term like risk management. In almost everywhere you will find risk management. Risk management means a lot of stress has been given on risk management. Then documented information. Every information has to be documented. You cannot hide anything. Then interested parties. It may be the laboratory, it may be the patient, it may be the supplier of the material, maybe the supplier of equipment or the consumable. Everybody is termed as the interested parties to align with modern quality management approaches. Then process approach. The new standard reinforces the process-based approach for managing the laboratory operations, emphasizing the importance of understanding interrelated processes. Means the process approach, it will be everything, whatever you are doing, that you will do for one, even like pre-analytical phase, you are working on pre-analytical phase, you will develop everything just keeping in view next phase also. It's not that you are doing something only for pre-analytical, no. There may be some adverse effect of your decisions on preclinical on examination or post-examination phase. Now, external services and supplies, here also a lot of stress has been given in 2022. There are updated provisions on managing external providers of services and supplies, ensuring that they meet the laboratory's quality standards. Particularly, this includes managing outsourced laboratory activities and evaluating supplier performances. If you say that we are comparing our results in interlaboratory testing, that we are comparing our results with Dr. Pankaj Kaul's laboratory. Now, the auditors, they will have the authority to check and audit Dr. Pankaj Kaul's lab also, along with me. Means whatever the supplies will be there, it may be consumable, maybe a process, maybe a report result, whatever, maybe equipment, full assessment of the supplier of that service, it will be auditable. They may ask for anything. Then environmental sustainability has also been stressed, while it's not a core requirement of 2022. Maybe in future when it will be revised, it may be a core requirement, but at present it's not core requirement. There is a recognition of the laboratory's role in environmental sustainability with guidance on managing environmental impacts, though through efficient resources use and waste management practices. Now, what are the major impacts of these changes? Global adoption and accreditation is first important or major impact. A wide adoption by medical laboratories has been done worldwide as a benchmark for accreditation. Organizations such as International Laboratory Accreditation Corporation, ILAC, and regional accreditation bodies like NABL support its implementation. This ensures global consistency in laboratory practices. Impact of alignment to 17102, sorry, this is a spelling letter mistake. It is 17025, one is extra. Now this provides consistency across various standards, making it easier for labs to implement an integrated management system. You need not to keep 3 different standards. Risk management approach, it has a lot of impact. The incorporation of risk-based thinking in ISO 15189-2022 represents a significant advancement in the standard, shifting the focus from reactive problem solving to proactive risk and opportunity management. Now, at present, what we do, we take just, we think incidents have occurred. After that, we take action. But in this, risk management will be first. Not more than 1 minute. Impact on healthcare. By providing a robust framework of quality and competence, ISO 15189 has significantly improved the reliability of diagnostic results, enhanced patient safety, and facilitated international collaboration in medical research and diagnosis. So I don't think a summary is required. I have elaborated everything. Thank you very much. Any question, please?
Any question from audience? कोई सवाल डॉक्टर मलकीत सिंह जी से? Any information? Yes, please. One question only, one and final. Yeah.
Thank you, sir. It was really a nice discussion on ISO 15189. So just to add one thing, that this is just to summarize the entire talk, that this is a most now in the refined stage, the standard ISO 15189. Earlier, a lot of disagreement between these standards were there. Now, most important thing which is we are now getting from this talk is that they have covered now point of care services also, which is a challenging area. Everywhere it's now, industry is moving towards point of care devices. So with reference to quality, it was a big challenge, but ISO 15189 2022 standard is now covering that also. And second thing is, of course, the risk management was there in the earlier version also, but this time they have more, have highlighted this thing, that risk management, even under the standard, there is a specific provision for risk management.
Actually, previously it was not separate. And previously it was discussed within the standard. Now they have given a separate title for it.
And one most important thing is that multiple standards were creating a lot of confusion between stakeholders. There is a generic standard ISO 1519000, which every organization is to follow. Then ISO 17025 is the second standard, which was earlier followed in lab also. So at the same time, few labs were implementing ISO 15189. So now they are trying to harmonize all standards together so that through one standard only, the lab can be accredited fully, covering all services. That just to add at this point.
Thank you. Thank you, Dr. Khan. Oh, please.
Quick question, sir. Dr. Monica from PGI. Sir, my question is that all the accreditation which was according to ISO 2012 is valid till 31st January 2024. From 1st January, all laboratories have to transit to it to 2022. The question is
See,
that,
sir, for the last few months, from July 1st, the new applications were being taken in 2022.
Okay,
But it is mandatory to be in from January 1st, 2025.
Sir,
After that, you have to change yourself.
sir, like certificates which were previously issued, they were till mid of 2025 for 2-year certificates which were issued. So for those laboratories, the internal audit and document preparation, this has to be done before that, sir. My question is.
See, your application will be going before that, but NABL does not have lot of staff. That is why they have given a transition period of 6 months. That those who have already got the certificate, they may continue till 25th, but no new application will be taken from July 1st, 23, July 1st, 24. Afterwards, there are applications of only 22.
Irrespective of the fact that we got certification in 2022.
Before that, you have to submit the reapplication for change for 2022. That is a transition period, nothing more than that.
Thank you, sir.
Sir, I would like to add on this. For the NABL accreditation, ma'am, if you already have a certification till mid of 25, so before 6 months you can apply for renewal of the certificate for new one only, and your certificate will be valid till 25 July itself. But if you want the new applications, you need to apply afresh if it will be expired. Otherwise, in that same login port, you can apply.
Before mid 25, you have to get new certificate as per 2022. Thank you.
Thank you, sir. So the next talk is by Dr. Pankaj Kaul. He is the Dean of University School of Allied Health Sciences, RBU Mohali. Sir, welcome you to the stage. The talk is on patient safety in clinical laboratories.
Good afternoon, sir. Welcome, sir. I'm Dr. Pankaj Kaul, Dean USAHS Mohali. Sir, please. She is, sorry.
It is all right.
Dr.
It is all right.
Kaul is a legend in medical laboratory sciences and a pioneer of the ICMLS and laboratory related associations, and he is very much involved in progress of the laboratory field. Thank you, sir.
Thank you very much. Thank you so much. सबको नमस्कार. This is the, I will say just professional introduction, but my actual introduction is I am a star centurion blood donor. 116 blood donations. I have donated my eyes, I have donated my body, I have donated my organs. And today whatever award was given to me, I have donated back it to my association with very gracefully. Because कहते हैं पेरेंट्स जो है अपनी बेटी के घर का नहीं खाते हैं, इसीलिए I have surrendered it, कैश वापस किया गया है. Okay, with this I will just start my topic. It is on patient safety in clinical laboratory. I have got a lot of pressure from my colleagues that earlier people of PGI have taken a lot of time, so I should not take much time. So, starting with this. Why safety in hospital? Hospital is a people-intensive place which provides service to sick patients round the clock. People have access to any medical treatment or advice. You can't stop anyone to come to hospital. Even if your enemy is there, it has to be handled properly. Safety of patient is therefore imperative to be maintained, especially in clinical laboratories. Patient safety in a clinical laboratory is about preventing errors that could harm patients and improving the quality of care they receive. Patient safety is a top priority because they are our customers. Patient safety has always been the top priority of all medical professionals, hence being knowledgeable enough in providing and promoting the right patient care and handling laboratory tests will avoid errors that could affect the patient health. That is why globally a commemoration day has been fixed, earmarked on 17 September when whole world commemorates Patient Safety Day. The theme for 24 was this year, improving diagnosis for patient safety, highlighting the critical importance of correct and timely diagnosis in ensuring patient safety and improving health outcomes. With the slogan, get it right, make it safe. Patient safety in clinical laboratory, each alphabet has got a meaning in itself. In safety, S stands for sense the error. A for act to prevent it, F for follow safety guidelines, E for inquire into accidents, T to take appropriate remedial measures, and Y for your responsibilities. You must try to understand that. So while dealing the infectious procedures, first of all we have to defend our own self. That is a priority. We must take full precautions of the safety gadgets which we are to use in the laboratory. We must promote a culture of safety. Medical lab professionals should promote a culture of safety that is patient-centered, timely, efficiently, equitable, reasonable, and genuine. There are so many factors which affect the patient safety in lab medicine, starting from, I can say, patient healthcare encounter, laboratory test, then come laboratory specimen collected, then laboratory tests analyzed, laboratory result reported, laboratory result interpreted, laboratory result reported to a patient, and then clinical action taken on actionable laboratory result. All these patient safety in laboratory medicine matters with each. Dr. Kalra has already explained all these things in the morning very, very clearly and very nicely through a beautiful PPT presentations, so I need not to go in much detail of these. So we must use quality assurance tools, as my colleague Dr. Malkeet and Dr. Surinder has already told. Implement tools to identify and prevent potential errors by using internal quality control and external quality control. This is a must. If you do not have any quality tools, you can at least review your own test samples which you have done internally, and with the side lab, you can take some sample from them and you analyze. That can be an internal and external quality control. You need not to waste much of the money if you cannot afford. So personal safety is the most important. As I have already told, you have to take full precautions and properly calibrate the things while you are working. You have to train your staff initially. Provide ongoing staff training and encourage open discussion about improvement, how we can improve. This is all what we are doing to improve our quality. On job, continuing medical education should be there. There should be workshop. There should be conferences like we are having, and if you go abroad, there are continuing professional development also. You do some workshop, you attend some conference, they are giving some points to you. Depending upon those points you have gained, you have achieved, they are getting promotions on the basis of that, or annual increments. So safe calibration should be there, and we must report our performance issues. Systematically, we must record and review performances issues as occurrences or incidents, as has been already explained in the morning by Dr. Kalraji. There should be a proper documentation of that, and there must be a proper record keeping. That is why this record keeping and documentation has been specially included in the model curriculum of medical lab sciences. Then, as far as lab specimen is concerned, we must ensure specimens are labeled with the patient full name, date of birth, and other identifiers so that the sample is not misplaced. As it was told in the morning by Dr. Lal also, that the sample was exchanged husband and wife. So, labeling is a must. We must follow safety protocols such as avoiding direct contact with chemicals, washing hands after removing gloves, and keeping the work area clean and organized so that next day morning when you will come, you should get a clean laboratory to work. The aftermaths are the errors which can have a significant impact on patient care. For example, a transcription error in a HIV result led to death of 3 transplant recipients in an incidence. Histocompatibility, organ compatibility, blood testing for blood grouping and all that, this is very important, otherwise the patient can die. So all these are very important aspects of this, and patient safety has always been the top priority of all medical professionals and must be dealt religiously. I am saying religiously, like you are very sanctily doing your puja, your gurbani and all that, your namaz you are doing, like that religiously you must work as a team. Thank you very much, and thanks, sir. Any questions, sir? These all things should be in our blood. We should not go in for protocol and all that, mentioned on the table or something. It should be in our blood. Everything we do, we should do it seriously and religiously. Then no quality control is required if you are doing it genuinely. I'll give you 100% guarantee that no quality assurance will be required. The same question I asked from Dr. Lal also. These accreditation and all that, they are taking a lot of time. And in most of the institute, nobody can deny we are short of staff. In labs also, in institutions also, sir. We are not getting full staff. The people who get retired, we don't get replacement for that. We are always short of staff. And then this paperwork increases our load of the work. And then what happens? The work which is actually meant for the betterment of the patient, that gets hampered. Dr. Malkit, yes, welcome.
Hello. No doubt time is not allowing, but Dr. Pankaj's call has touched a very, very important topic. What I would like to say, that whether we are accredited or not, our positive attitude and working with concentration is very, very important. Working honestly is very, very important. I will just tell you one example, you will learn just what I wanted to say. Once my brother's son, he was sick and he took his sample, one of the charitable labs. मैं इस पे बताऊंगा कि charitable क्यों बोला. उसका रिजल्ट आया CPK का, रिजल्ट नॉर्मल, और उसका he was a declared case of Duchenne's muscular dystrophy. PGI में उसकी वैल्यू आ रही थी 3000 प्लस CPK की, वहाँ पे उन्होंने दिया 25. और ब्रदर मेरे को जैसे सुबह बात हो रही थी कि उसने मेरे को कंग्रेचुलेट किया कि अपना बेटा तो ठीक हो गया. I did not agree to that कि ऐसा नहीं है. मैं 3 दिन बाहर था, 3 दिन में 2 बार टेस्ट कराया, दोनों बार about 25. When I reached there, मैं गया उनके लैब वालों के पास, उनसे बोला कि आप मेरे को सारा टेस्ट दोबारा करके दिखाओ. उन्होंने तीसरी बार टेस्ट किया, almost same report. When I checked their kit, just by the name of PGI, मैंने उनकी किट्स निकलवा ली. All the kits were expired. All the kits were expired, and the persons were there. They were working with expired kits. Probably these charitable working people They don't spend money. Somebody must have given them a month expiry date or 2 months expiry date. Those expired and they're using, continuously using, saying this is just a donation, charitable, it is charitable. But when fresh kit was used, his value was 3,600. So who is responsible for this? Charitable hospital? Or I will say, the person like me who was working there, he or she is responsible. Why did she not object to it, that I won't use these such kits, expired kits? So we have to be honest on our seat, only then our profession will get respect, our cadre will get respect. Thank you very much.
On time, sir, you completed, and it is very important also to know about this. Thank you, sir.
Thank you, ma'am.
Thank you, sir. The next talk is by Dr. Tirumurugan Murali P. He is the principal, College of MLT, MT, PG and RIHS, Puducherry. He is going to speak on career in academics and research, unexplored potential of MLS professionals. Welcome, sir. I request Ms. Maliga to kindly introduce him.
I am very glad to introduce the speaker of this session, because I am working under him, and why I am today is also because of him. So on board it was written many things, and one thing I want to tell is he works for the student welfare and betterment, always things for the students, and he will remain as a teacher. And every student who have finished and completed, settled, and they will think of him, and very happy when all the people see him and says that because why they are now, so it is because of him and his guidance, everything, and still he is doing what can be done for the betterment of the students, and with the health department of Puducherry, and fighting for the students to get into the government jobs in our setup. Welcome you, sir.
Thank you, Balika, for the introduction.
Sorry for interruption. Actually, just an announcement after this. This is the last talk of this plenary session. So after this, we will have two panel discussion, very interesting panel discussion. One will be on NCHPF. Those who are outside, please also ask them to come inside, because NCHP is the core of this conference now. The entire country is now following that NCHP regulatory frameworks. And second panel discussion will be on cadre allowances and career pathway also for the student also. It's going to be a very interesting session. So both sessions are very important. We have experts from the industry, we have experts from association, we have the leaders from the various INIs and very prestigious institutions. We have experts from the academics. So please join this and ask your friends, colleagues and students to come inside, because just two panel discussions will be there. Tomorrow the entire day will be full of scientific discussions. Thank you. Sorry for that.
Thank you, sir. From morning we have been hearing to all core subjects of medical laboratory science. This will be slightly different. That's not core subjects, but more to do with what we are expecting to happen in the profession of medical laboratory science from here on. Usually in my college we have a speaker called Dr. Gopal. We all wish that we speak before him, because once he takes the stage, almost everything is disseminated. So next time I think I should talk before Dr. Kaptan Singh Sharabat, because morning as he delivered the keynote address, almost everything that I prepared has been told in that, but I'll try to make it very simple and interesting here. So the journey so far, we know that science evolves, it coexists. Lenses Microscopes came, then we know about microbes and cells. Without computers, the Human Genome Project could not have been completed. So it is always coexisting. So has been medical laboratory science. It's the techniques and technology that evolved, and the profession also grew along with it. I'll just take you through how we came over here. We'll see the technical revolution, how it happened. It'll be very fascinating to see her as long back as 1620. You can imagine how many years back. This verbatim will produce those words there. We also have glasses and means to see small and minute bodies perfectly and distinctly, the constituents of blood and urine which are not otherwise visible. These words are documented in a published document by Francis Bacon, that is in 1620. You can imagine how long it is. But the evolution of technology did not happen that fast. It took almost 200 years till we came to a position where that could be systematic analysis of blood and urine. So the development phases can be grouped. I'm not dwelling into the details of it, but all this has got to do, initially there were no separate laboratories. The scientists were working, they used to have their own labs in their places, and they worked on it. Then slowly as technology improved, then it became institutionalized. Now they had hospitals, then the research labs, etc. came up, and also a phase of extension when really machines came into it. So there are two major contributions which actually contributed to this. One is the miniaturization of the entire instruments as such. You'll be surprised to see that the centrifuge that was first designed, it was put up in a 10 by 10 feet. Imagine how much amount of samples it could have taken. So once the miniaturization of the instruments came up, that did away with the contradiction between the quantity of the samples and the limit of detection. The other thing is the word which we have been hearing for obtained number of times from morning. It's about automation, and that resolves the contradiction between speed and precision. So this is how it has been moving. The transition of technology, it was tools to machine, and now to automation, and now we talk about artificial intelligence and all those things. So the paradigm shift has been laboratory automation technology, which you have been hearing, molecular diagnostics and personalized medicine, robotic laboratory assistance, artificial intelligence and data analysis, and of course the upcoming field of wearable health monitoring devices. So this is how technology has evolved itself, but has the medical laboratory profession also has come along with it? We'll see. So to provide human resources to keep up with pace of this improvement in technology, that has to be supported by a good academia. Initially, there were not any structured curriculum or courses that were being offered. In fact, it was an unstructured training. If I could remember, I heard from people, my seniors, that even in JIPR, there was no courses that were there exclusively designed for medical laboratory professionals. There was a training that was offered. In fact, there was a circular given to all the people who were working in JIPR, even in offices they were there. So a circular was sent, and it was, an undertaking was taken from them. Whoever wants to join and work in the laboratories there, they could come forward. So they were given a training, and all of them became laboratory technicians. So it was at that point of time. But we know it is always a necessity that will make things happen. So